This method can help us answer key questions related to gene expression, showing us exactly how chromatin structure impacts gene expression and how chromatin organization regulates transcriptional dynamics.
We developed this technology because we wanted to be able to controllably alter chromosomal architecture to enable creation of long-range chromatin looping, while at the same time have the ability to reverse the chromatin context to restore the endogenous chromosomal structure. This method was designed for ease of use and wide applicability. We took advantage of a non-toxic dimerizer as well as orthogonal species of dCas9 in order to allow the system to be used more broadly. We developed this method because we wanted to be able to answer the long-standing chicken and the egg question of whether gene expression begets chromosomal architecture or whether chromosomal structure leads to changes in gene expression.
Design of the CRISPR guide RNA sequences and maintenance of the cell cultures are described in the text protocol. Plasmid maps and the primers utilized are listed therein. Begin the plasmid preparation by mixing 5 micrograms of the lentiviral CRISPR plasmid with 3 microliters of BSMB1, 3 microliters of alkaline phosphatase, 6 microliters of 10x digestion buffer, and 0.6 microliters of freshly prepared 100 millimolar DTT. Bring the total volume to 60 microliters with double distilled water and incubate the mixture at 37 degrees Celsius for 30 minutes to digest and dephosphorylate the plasmid. Then, gel purify the digested plasmid and elute the plasmid in double distilled water. Next, prepare annealing reactions for the guide RNA pairs.